So I think we'll get going. My name is Michelle Bayeux, and I'm just going to get us going with the cluster presentation. So welcome for those of you that haven't attended the sessions before. Thank you very much for attending this Realizing Healthier Pregnancy Research Cluster presentation. We're so pleased to welcome you and thank you very much for joining us today. My name is Michelle Bayeux, and I am the Research Grants Director at the Women and Children's Health Research Institute, and I'm pleased to serve today as the session moderator. On behalf of WCHRI, thank you so much for taking the time out of your very busy day to join us. I'm just going to share our slides for acknowledgement, to do a land acknowledgement. So this session is taking place on Treaty 6 territory, the traditional lands, traditional lands of the Cree, Dene, Nakota Sioux, Saulteaux, and Blackfoot Nations, and the homeland of the Métis. We would also like to thank the Stollery and the Alberta Women's Hospital Foundation for their generous contribution to WCHRI, which supports this cluster. Just let me go back to my, sorry. So I would like to just remind you that WCHRI is actively seeking to build the research cluster. So please reach out to WCHRI at wcgrants@universityofalberta.ca to nominate your collaborators or networks. You can also contact us directly if anybody would like to present. We'd love to hear from you. So just drop us a note and we'll include in the chat the grants address so that you guys can help facilitate you guys contacting. This session is recorded and will be posted to the WCHRI website. We'll send out a link to the recording about 24 hours after the session is over. If you would like to share any of your contributions, we look forward to questions, comments, concerns after each presentation. So on to the main event, I would like to introduce to you our two presenters for today. Anna is going to be a little bit late, so we're going to start with Dr. Rachel Tribe. So Dr. Rachel Tribe is a professor of maternal and perinatal sciences at King's College in London. Professor Tribe's research focuses on translational research related to preterm birth and other pregnancy-associated conditions. Specific interests include developing prediction tools for preterm birth, host microbiome interactions within the female reproductive tract, and uterine smooth muscle physiology. She also collaborates with colleagues to study the impact of pregnancy exposures on the development of the neonatal immune system. Dr. McKinnon, she's not in the space yet, but I'll just tell you what her research interests are, and then we will reintroduce her once she is available. Dr. Anna McKinnon is a clinical psychologist and assistant research professor in the Department of Psychiatry and Addiction at the University of Montreal, and scientist in perinatal mental health and early child development at the CHU Saint-Justine Israeli Research Center. Her work aims to identify risks and protective factors to inform the development of innovative programs that promote resilience and prevention of intergenerational transmission of distress. So we're going to now ask Dr. Tribe to take the floor.
Thank you. So can you see my slides? Yeah, great. I'll just get a pointer. Right, so thank you very much to Michelle for very kindly inviting me to speak at this session. it's a real delight to see so many people that online. I'm sitting in sunny London. It's late evening, so I apologise if I'm a little bit jaded, but I'll do my best. And I was thinking about what I should talk about today. I'm a translational research scientist who's trained in physiology but done clinical research for most of my life. So I thought I'd give you a sort of a feeling of the work that we do here at King's. We work from the molecular to the bedside. We've always got a focus on how our research can actually have impact. And I think that gives us a little bit of a unique flavour to our research activities. So having said that, I'd like to say thank you to everybody who I work with, because doing translational research in the clinical space is incredibly difficult and hard work. And it actually needs a whole family of researchers to work together in an interdisciplinary way. And this slide just shows you a host of the scientists, the bioinformaticians, statisticians, immunologists, clinicians, biochemists that work in my group mainly over here. And then the clinical team, my long-term collaborator, Professor Andrew Shannon, who's a consultant in obstetrics. And this clinical team is absolutely fundamental to the research that we do. They help us recruit women from the preterm birth clinic, and we work hand in hand. And, you know, the women generate the questions, clinicians generate the questions. We try and think about how we can answer those. And then we have teams that I work with in Africa and India, and also a team that I work in doing some uterine mapping, which I'm not going to talk about today. So for the purposes of today's talk, I'm going to talk a little bit about preterm birth, the problem and the challenges. I assume that many people on the call will know these, so I'll try and keep that short. And then I want to talk to you about a pregnancy cohort that we set up back in 2014 and give you a flavour of how we've used this cohort to do some science, to do some biomarker discovery for prediction of preterm birth, and then move on to show you a little bit of what we're planning to do in the future in case anybody would like to collaborate with us. So for those who are not working preterm, but in the preterm birth space on the call, preterm birth is less than 30, when babies are born less than 37 weeks of gestation. And it's a huge problem. Just looking rather nationally at England and Wales, where preterm birth rates are 7.9% last year, which is a rise from 7.5% the year before. So that it's still a problem in many countries. Most sort of global north countries have rates similar to this, and I looked up Canada's preterm birth rates, and I think it was about 7.8, so very similar. But the burden of preterm birth, you can see here, is also predominantly held by the global south, and rates can be up to high as 20%. Some of the work that we've been doing in India has actually shown with accurate gestation dating that preterm birth rates in rural India are up to 24%, which is just awful, really. And why is it awful? Because babies die when they're born preterm, and if they do survive, then they survive with high rates of morbidity. These can be acute at birth, which can confer even more risk after birth, and also there's long life implications of being born preterm. So we have to try and prevent it. And the problem is that prevention and treatment options are very, very limited. And part of the reason for this is because spontaneous preterm birth, which accounts for nearly 2/3 of all preterm births, is actually a syndrome. There's lots of influencing factors, and there's lots of pathways that lead to the common presentation of a pregnancy ending in a preterm delivery due to premature shortening of the cervix and the initiation of uterine contractions. And you can see here that if you look on the left-hand side, there's a whole lot of exposures and influences that can increase a woman's risk of having a preterm birth. A woman comes into pregnancy, and every pregnancy she comes into, she's different, because she's influenced by the pregnancy she's had before. She's influenced by getting more morbidities, comorbidities as she ages and has more children. Her BMI may change, her health just generally may change. So we have to think about personalised medicine for women who are pregnant to understand their risks. Some of these risks are modifiable, like the nutrition and exposures and behaviours, but some of them are not. And so that includes obstetric history, maternal genetics, and maybe some uterine and cervical disorders that a woman will bring into a pregnancy. The pregnancy itself will also be affected by the fetal genetics, so therefore every pregnancy again is going to be different. And then on the right-hand side, you can see here that these are the lots of different pathways that lead to preterm birth. And what we've been trying to do is find a single drug and a single test that predicts people's risk of having a preterm birth. But you can understand it being a syndrome, that's going to be very difficult to achieve, and actually is half the problem why preterm birth rates haven't changed in the last 25 years, really. So what do we have in the clinical arena in our toolbox that can actually help us managing preterm birth and risk in a pregnancy? Well, I've got two women here. The green woman represents a low risk woman. This is generally somebody who's had a previous term pregnancy or somebody who is in her first pregnancy. And the purple woman here represents a high risk woman who, based on her medical and obstetric history, is already at high risk of preterm birth when she comes into pregnancy. Unfortunately, we can do nothing for the low-risk woman in pregnancy. The first time we'll know she has a risk of preterm birth is when she presents in premature labour. High-risk women we can do a little bit more for, and these are all the tools, and it's shockingly small, the number of tools we have to look after pregnant women at risk of preterm birth. We can monitor their risk, and there are two main tools that we use in the UK and Europe. They're not necessarily both of them available in Canada or in the US. Our first tool is a swab for fetal fibronectin, and the swab is taken in the cervico-vaginal area. And fetal fibronectin, when it rises, is an indicator of increased risk of preterm birth. So we can measure that and get a quantitative value for it. And we can also measure the cervix, and this is a transvaginal ultrasound you can see here with the pink arrows, and we can measure the length of the cervix. And a woman's risk of preterm birth increases as her cervix shortens, and as it shortens less than 25 millimeters, the risk is considered high. And we can put these bits of information into an app, which we call the QUIP app, and I haven't got time to go into that, but we have, as a broader team, developed this app for managing risk, talking to women about their risk of a preterm birth. But if somebody does get a short cervix or seems at high risk of preterm birth based on our app, then we can offer 3 interventions for a short cervix. One is putting a stitch in the cervix to hold it shut. One is giving vaginal progesterone, or there's another device called the Arabin pessary that closes and holds the cervix shut. But unfortunately, there isn't a huge amount of evidence to say these are really efficacious, and none of them actually improve outcomes for the baby itself. And then the other few drugs that we have for treating preterm labour are when women present with contractions, and we have tocolytic therapy, which have most of those, like nifedipine and etosaban, whilst they might stop contractions, don't really confer benefit to the child. And we have these sort of tools in our armoury to be able to give women corticosteroids to mature the fetal lung if she presents in threatened preterm labour, and to give magnesium sulfate to protect the fetal brain. But that's all we have. And you look at these tools, and most of them are focused particularly in the preventative space, focused on the cervix. And from my slide before, I showed you that there's lots of routes to having a preterm birth. So the tools we have may not fit everyone who's at risk of preterm birth. So what can we do about it? So back in 2014, we realised that we didn't really understand the natural history of a pregnancy that was destined to have a preterm birth. So we set up a long-term longitudinal cohort recruiting women at risk of preterm birth and monitoring their biology. And we've created this very good biobank and database of clinical data that allows us to really dig deep into understanding what the biological mechanisms are for the reasons why a woman may be having a preterm birth. And just to explain to you what we do, we've recruited over 1,000 low-risk women. We see them at 2 times in pregnancy. It's very pragmatic because they come in for a nuchal scan around 10 to 13 weeks, and then they come back between 20 and 23 weeks for anomaly scans. So that's when we can capture the low risk women, and we can capture them at delivery too. For the women who have a known risk of preterm birth, and this is because they've had a previous preterm birth or PPROM, which is preterm premature rupture of membranes, or they have had a mid-trimester pregnancy loss, they're deemed high risk, and we can follow them in our clinic every 2 weeks and take samples and gain clinical data from them. And we take a range of samples from both low risk and high risk women: blood, buffy coat, vaginal swabs. We take a cytobrush, which is a cervical swab. And then for high risk women, we can measure that cervical length and fetal fibronectin as they come for their visits. So that's the cohort. What can we do with it? So the work that I'm going to talk to you about today is mainly focused on understanding the cervicovaginal environment and how that confers a risk of preterm birth. I'll move on to also thinking about other biomarkers and other risk factors later on. But you can see here we have the cervix and we have the vaginal environment. And within that environment, there's a resident microbiome, and that there's there's a lot of host defense going on in the vaginal environment to try and maintain a healthy environment. And a healthy environment, according to the literature and some of the work that we've done, would suggest that the bacteria in the vagina is predominantly Lactobacillus crispatus, which is a benign bacteria which promotes a very low pH in the vagina, which excludes other bacteria that may not be so benign. These bacteria produce lots of lactate, and so you've got a low level of host response. It's not a huge inflammatory environment. You've got elafin, which is a natural antimicrobial peptide that's also protecting the vagina from nasty bacteria or viruses or other pathogens. And so when you're in that environment, we think even if the woman has a risk of preterm birth due to other factors, this will keep her cervix very closed and long, and then protect basically the baby inside the uterine cavity. But what we predict is that when women have a more diverse microbiome, and this is made up of mixed anaerobes, that actually they have a higher pH in the vaginal environment. They're producing a whole host of different metabolites, so it shifts from being lactate to being more acetate driven. There's more inflammation, and there's attraction of neutrophils to this space. And we think that the inflammation itself, in response to this bacteria, is affecting the cervix, and the cervix will then shorten, and then there's a risk if there's any nasty bacteria within the vaginal microbiome, these can ascend and enter the uterine cavity, causing even more inflammation, and that in itself can trigger preterm birth. So that's what our hypothesis is, and we have data to support many areas of this hypothesis. So I'm going to show you a few of the studies that we've done so far. So this is the power of having this cohort. We had to collect it for a long time before we could start doing analyses on it. But this is one of the first big studies that we published from it. And this was looking at the vaginal microbiome in 346 women. 240 women had had a previous preterm event and were high risk in the pregnancy, and we had low risk women of about 100. And we have a mixed population in London, which is good because of diversity and ensuring that whatever our research we do is actually relevant to all women, and it's not just on a very highly defined population. And what we did with these 346 women, they provided us samples at 2 time points in pregnancy, and we analysed their microbiome using 16S sequencing. And we also looked at the metabolites that were being produced with the specific microbiome that we were measuring, and we did that using NMR. The details don't really matter, but it meant that we could see the metabolites, we could see the microbiome, and alongside that we measured some antimicrobial peptides. From that previous slide, elafin we think is promoting a healthy environment, and cathelicidin, which is predominantly released by neutrophils, is actually an indicator of a more diverse, more inflammatory environment. So I'm not going to go too much into the details, but just try and bring you along with the story, because we're looking at trying to see whether we can harness this information to predict preterm birth early in pregnancy. So we've measured the microbiome, just so that you know, we could identify 5 communities of bacteria that were represented the women that we were studying. You had a group, and these colours I will show you in all the slides. So purple is Lactobacillus crispatus, that healthy bacteria. Then you have pink, which is Lactobacillus gasseri. You have Lactobacillus innes, which a lot of people think is a nasty microbiome. I suspect it's actually protective for reasons that we can talk about later if you're interested. And then you have a very mixed microbiome in this light blue, and then you have a slightly mixed group here, which is crispatus and gasseri. Now we've looked at this, and we can tell that there are differences in the communities and risk of preterm birth. So as I mentioned earlier, if a woman is high in Lactobacillus crispatus, this microbiome produces lots of lactate, and the risk of preterm birth is generally low. And you can see this relationship here. This is just to illustrate the sort of power of what we can do with this information. And we can see here how different metabolites relate to different groups of bacteria, different communities that we've detected. You can see here lactate is negatively correlated with the diverse microbiome, and acetate has the reverse relationship. And we've measured lots of metabolites, and if you're interested, I'll direct you to the paper to look at that in more detail. We've also looked at elafin and cathelicidin, and as we hypothesized, elafin, this antimicrobial peptide, is associated with a healthy microbiome. And cathelicidin, when we think that as you get more dysbiotic, there's more neutrophil invasion, there's actually more cathelicidin produced in the vagina. So we're getting a feeling of what this vaginal environment is like in women, and so it's evidence-based rather than hypothesis. But what we wanted to do with all this information is actually integrate it and say, can we develop a biomarker to predict who's most at risk in early pregnancy? The earlier we can identify somebody at risk, the more likely we are to be able to intervene and modify her risk. And actually, by understanding what's driving that risk, we can also start thinking about potential treatments. And this slide is just to show you the basic way we integrate the data, and we've done a lot of machine learning around this and various studies. But the upshot is that we're trying to develop algorithms that can predict preterm birth. And this is just an example of the sort of work we do. These graphs are showing ROC curves. So it's the area under the curve that gives you an indication of the ability of these combinations to predict preterm birth. We started looking at all the metabolites because we thought that actually people have lots of different bacteria, but the metabolites might give you a composite view of what that community is like, because it's the sum of all of those bacterial activities. And we came up with a composite with a number of metabolites in and looked at its ability to predict preterm birth. And for those who are not familiar with ROC curves, we're looking for something that's above the line of chance, which is here, which would be 0.5. And we're looking for ROC curves that go up towards this corner up here and here. So you'd have perfect prediction if it was a square. So here we have a prediction, a metabolite test, and its prediction is about 0.75, which is actually very good for these type of prediction tools. And we would imagine that we would use something like this for screening women in early pregnancy and getting an idea of what her vaginal environment looks like. Is she somebody at risk because of her vaginal environment? And we've also tested to make sure that this test works on all of the women in our clinic. We've normally looked at self-reported ancestry, so women report whether they're of Black ancestry, white ancestry, or other, and we can see here that this metabolite test works well for all of these ancestry reporting groups. We don't think that necessarily this is what's driving the vaginal microbiome. It's probably a surrogate, and I'll talk to you a little bit more, because I think this is quite a crude way of looking at it, but it does make sure, particularly where there's disparity in healthcare, that we are actually making sure we're developing tools that are suitable for everybody. This I'm showing you is another prediction where we've actually tried to introduce the use of bacteria. And to cut a long story short, we have differences depending on ethnicity. And the problem is that in white women, if they have lactobacillus crispatus and it's dominant in the vaginal microbiome, they're very unlikely to have a preterm birth. But you can't say the same for black women. And so when we've tried to develop prediction tools just with microbiome, they don't work. They work slightly better if you combine them with metabolites. But the graph on the right-hand side, you can see here in the orange line, that's the composite prediction. It's 0.758, so similar to the metabolites, so potentially useful. But it works better in white women and less well in black women. So we have some work to do around developing that type of test for prediction. We do, however, think that we could use the lactobacillus crispatus screen as a sort of screen just at the beginning of pregnancy, but with the awareness it's not useful in everybody. And just to illustrate this on this slide, just look at the colours. If I was showing you this slide for the women who reported white ancestry in our cohort, you would see that predominantly all of the term women were high in lactobacillus crispatus and the preterm birth women were dysbiotic. But you can see here, and this is just for women reporting black ancestry, that there are a whole range of vaginal microbiomes associated with term delivery. And so lactobacillus crispatus, which is in the purple, isn't the major predictor of outcome for when you're looking at microbiome alone. And that's something we're now considering and trying to refine. I don't necessarily, I think there may be there's other issues at play here that may be due to diet, maybe due to socioeconomic status, BMI status, age. There's a lot of other factors that could explain this. But it has driven us to really think about if the vaginal microbiome is important and driving preterm birth, that it's not as simple as just classifying the bacterial communities into these 5 states. And so we've been doing some bioinformatics to try and understand the communities within the communities, what bacteria live together, and are they more likely to cause problems if you say got Gardnerella vaginalis and you've got Prevotella and then you've got some other bacteria within that community. Is that what's putting a woman at risk, rather than just broad brush strokes with the sort of 5 microbiome communities we've described earlier? And actually using this type of approach, which is has been very useful, we're now doing it on a second cohort and really doing a deep dive, and we have identified some key bacteria that are associated with preterm birth risk in all women. And we can use that type of data. These colours at the side represent those clusters I've just shown you. And we can start looking at how those clusters are influenced by more than just ethnicity. And so you can see here that in the red, that's associated with the clusters, and these are different bacterial clusters that I showed you on the previous slide. And you can see whether those clusters are not related to certain activities. And just to highlight, you can see that if the metabolites putrescine and succinate are highly associated with the bacteria that we can see in these communities and other metabolites. And they're also associated with a higher pH and higher levels of acetate. So you can start dissecting these individual clusters of bacteria to understand what their function is and what they're doing. And what's interesting is these clusters are predominantly associated with a low gestation at birth, i.e. preterm birth, and they're also associated with a high BMI. And what we're beginning to see is that BMI, which we know confers a sort of inflammatory state on the woman, is also influencing the vaginal microbiome. So BMI in itself is potentially mediating its effects on risk of preterm birth through modifying the vaginal microbiome. We're also looking at the cervical immune cells, because obviously the microbiome and the response of the cervical immune cells are very important. There's a relationship between these. This work is incredibly difficult, and all of this work was done with fresh swabs from pregnant women. We're getting an idea of the profiles. We know that neutrophils are high, and that there's high T cells and B cells are potentially implicated in what we're seeing. But this is a work in progress. It takes us a long time to collect all these samples, because we're collecting these early in pregnancy before we know the outcome of the pregnancy. So it's all prospective, but we were getting some really good ideas of what's going on. I won't go into this too much, but we can actually look at the gene activation in neutrophils in response to specific bacteria. And again, we're getting a feeling for which bacterial combinations are causing an inflammatory response, and we hope that will feed into what we're doing. Just as an example, we're now taking everything that we've done in vivo, and we're starting to do experimental work in vitro using a vaginal epithelial 3D culture. And we can grow multi-layers of vaginal epithelium and get them to differentiate and start producing mucins and glycogen. I'm not going to show you any data from this, but if I were to speak again, I could tell you about the work we're doing with co-culture with bacteria and neutrophils in this model, and it's really useful for going backwards and forwards between your clinical observations and then looking for your mechanistic observations. So just to sort of end on the last 3 slides, this was just to give you a feeling of what we do. How are we going to use this knowledge? Well, we're going to try and operationalize it by thinking about bedside metabolite testing, bedside bacteria testing perhaps, but we're more interested in rapid sequencing of bacteria. So we're working with Oxford Nanopore to develop protocols that we can use in our clinical testing labs here in the hospital, so that we can test a woman at one visit and the next visit tell her what her vaginal environment is like. We have this app that we've already developed, so hopefully we can integrate this into the QUIP app and start providing more personalised prediction based on more knowledge. We're aware that we're not just looking at the cervical markers, and so we're doing some work on plasma and serum and saliva and hair, which I don't have time to go into. But just to say that some of the work that we've been doing with the commercial collaborator, Mervi, a spin-out company from Stanford, is that we've been identifying some markers in the plasma as well, using cell-free RNA. Again, I won't go into it in too much detail, but we found 25 cell-free RNA transcripts that, when you put them together, are good at predicting preterm birth. And it's quite interesting that if you then look at the proteome in the same plasma sample, that you can also find protein differences, and we started combining these data and we're getting even better prediction of preterm birth. And we're thinking that we can use a variety of tests and combine them to be able to predict preterm birth in individual women who have individual causes of their preterm labour. We have to make sure that all of our work is relevant, so we're currently working with the PRECISE network on a study on spontaneous preterm birth, and we're looking at pregnancies we're looking at all the preterm births from a cohort of 6,000 women, and looking at their vaginal microbiome, their metabolites and their plasma proteome, and looking at whether the predictors we have are useful for women who live in the Gambia, Kenya and Mozambique, because we want to make sure, as I said, that our tools are useful for everyone. And then finally, just to say that we now have a new Insight 2 cohort, which is following pregnancies and also into childhood. And we have funding to follow children up to 24 months. And this allows us to ask a lot of questions in pregnancy. We are focusing on preterm birth still, but we're also going to be recruiting women who are at risk of preeclampsia, fetal growth restriction, gestational diabetes. And we've just received funding from the Helmsley Trust to look at how pregnancy exposures actually increases risk of type 1 diabetes in the child. And we started this whole cohort back in October. We've recruited about 100 women so far for this. But if anybody's got any questions on this, I can tell you what we do in more detail. So I'll leave it there just to say that we're trying to use our science to develop biomarkers to predict preterm birth. We want to understand the phenotypes, we want to understand the risk for individual women, and hopefully our approach will allow us to do that. Thank you. Oh, and I've got this picture. I'm sat in this little building here. That's the hospital, and we overlook Big Ben.
Thank you so much, Rachel. That was a wonderful presentation. Can I ask if anybody has any questions to get us going? A couple of pages. Let me just go. Alicia, can we please ask you to ask your question?
Yeah, I'd love to. Great presentation, Rachel. That was so interesting. Thank you so much. I had a question about your gene activation data you were showing in neutrophils they were exposed to bacteria. Was that correct?
So these were taken from cytobrushes from women, and then we rapidly isolated the immune cells and cell sorted them and got a neutrophil enriched population, which we then extracted RNA from to do bulk RNA-seq. But we could then use bioinformatic tools to get clusters of activation of the neutrophils and align it with the microbiome that we'd co-measured using metagenomics, and the same women at the same time, 2 different swabs. But yeah.
Wow, that's fascinating.
Yeah.
Do you anticipate that any of these markers of activation might also be observed in some of the neutrophils in the periphery?
Well, yeah, that's an interesting question. I've been writing a PhD application this afternoon, which is to actually carry on the work in the cervix, but also to collect maternal blood to do the same. We have done some mass cytoph work trying to compare the cervical cells and the circulating cells, but we want to do something that's a bit more high throughput. So yeah, we're going to start doing that.
Amazing. Thank you.
Thank you so much, Kalisha. William, do you have a question?
I do. Trying to both unmute myself and make myself visible. Hi, thank you, Rachel. That was great. And I like your picture at the end, because I spent a lot of really just terrific and informative and exciting times at St. Tommy's.
Oh,
people both in the preterm nursery, Tom Ariki and others, but also you have such a great team that I'm sure you're exploring, as it looks like you're just making a wonderful profile of a number of different potential both biomarkers that are going to be predictive, but also give you information about mechanism. So in that long-winded intro, I'm wondering if 2 things. I think you alluded to maybe the group you're working with in San Francisco or something, where extracellular vesicles may also provide kind of information as well that adds to this profile. I'm wondering if you ponder that. And the other one is, in the world where now we can do so many more things about measuring various maternal phenotypes at home. The one I'm thinking of in particular is heart rate variability and autonomic nervous system stuff, because there's clearly a story there about prediction of preterm birth. So I'm wondering if you've pondered adding those to your armamentarium.
Yeah, so extracellular vesicles, definitely. We had actually funding to look at cervico-vaginal extracellular vesicles. It's actually quite a difficult thing. We spent 2 years trying to isolate them and characterize them, and it was difficult, I would say. In terms of the maternal circulation, obviously that's going to be an easier ask.
Right.
And we did actually start doing that with collaborators in Australia. And actually the work that we're doing with Mervi, we think that the vast majority of the cell-free RNA is actually coming out of extracellular vesicles in the maternal blood, because the kit they use is actually for extraction of RNA from those vesicles.
Oh, that's pretty good. And the heart rate one, have you pondered it all, or is that just?
Yeah, well, we've got a new head of school that started last October, and he comes from Singapore, and he's very interested in digital health.
Yeah.
And so we've been talking to him since October about putting wearables on our women so that we can look at heart rate variability, not only for prediction of preterm birth, but some other sort of obstetric events.
Right.
It's just a matter of getting the money.
Sure.
And also the governance around monitoring where somebody lives is with GPS and everything is, and then working with a company to do that is really tricky. We've just spent 3 years trying to do something with GPS tracking and air quality with backpacks with women, and that was with Dyson, and it was, the governance was really hard to get over. We've managed to get over it, but Dyson's closed down their air quality backpack measurement program, so we've had to find something else to use. But yeah.
Well, good. But now there are little devices, you know, like wristwatches and all, and we've dealt with this with large cohorts around the world. So maybe I'll reach out to you later and see
Yeah,
if
that would be
there's any
great.
use to you.
Yeah,
Okay.
yeah.
All right. Thanks again.
Thank you.
Thank you so much. Does anybody have any questions for Rachel? So I'm going to thank Rachel so very much for her presentation. And of course, she will remain in the space while Dr. Anna McKinnon presents her work. So as a reminder, I'm just going to, Dr. Anna McKinnon is a clinical psychologist, assistant research professor in the Department of Psychiatry and Addiction at the University of Montreal. She is a scientist in perinatal mental health and early child development at CHU's Saint-Justine Israeli Research Center. And her work aims to identify risks and protective factors to inform the development of innovative programs that promote resilience and prevent the intergenerational transmission of distress. So Anna, if we can get you to take it away, that would be lovely.
Okay, thank you for that kind introduction. Can everyone hear me now? Yes, okay. We'll move over to something psychosocial. Before starting, I'd like to acknowledge that I'm joining you from Montreal, or Tiohtià:ke, which is situated on the unceded Indigenous lands of the Kanien'kehá:ka Mohawk Nation of the Haudenosaunee Federation. I'd also like to offer a reflexive position statement as awareness of my privilege and biases. So I'm weird-ish, a white cisgender woman, first to earn a PhD in my family, single with no children. However, I've had the pleasure to work with hundreds of families from all different backgrounds through research and clinical practice, and this contributes to the lens through which I work. I'll make a quick note on inclusive language as well. Most perinatal research has focused on participants who are assumed to identify as cisgender women, and while their experiences are important, we don't want to exclude gender diverse folks. So I'll also use the terms, we'll use the terms maternal and women sometimes if that's the language that was used for recruitment or reported. And when that hasn't been specified, or to be more inclusive, I'll also use pregnant persons or birthing parents. So my research program starts with epidemiology. I use that word loosely, looking at patterns and determinants of health, so perinatal and new parent populations. And I'm going to highlight a few studies that point to potentially modifiable risk and resilience factors for early child development to inform treatment targets. And we use bioecological models to study the developmental origins of health and disease, even in mental health and development. And this suggests the need to examine multiple embedded or nested levels of context within which children develop. So first we'll talk about some findings at the individual and family levels. When I was a grad student many years ago at McGill University, my dissertation involved a follow-up study of about 340 participants from pregnancy to 3 years postpartum. And then we invited 188 of them to come back into the lab with their 2.5-year-old toddlers. We filmed 5 minutes of free play, and that was coded for parent behavior. We administered a battery of 3 theory of mind tasks to assess early developing social cognitive abilities. And we also took saliva samples from the kiddos, and that was used to measure oxytocin receptor gene methylation, which is an epigenetic measure or proxy of gene expression. And what we found was a significant interaction for structuring. So that's supportive and appropriate organization of child's play. If we look at the side of the graph here where methylation is high OXTR, where the oxytocin system is presumably downregulated or not functioning well, we see worse theory of mind performance when structuring is low, the solid line, but exposure to high structuring, the dashed line, moderates this effect. So these results suggest that maternal parenting behavior may buffer the effects or be protective of downregulation in the oxytocinergic system for children's early developing theory of mind abilities. So this is potentially a modifiable factor number 1, parenting. And then as a postdoc at UCalgary a few years ago, I had the opportunity to use the All Our Families cohort data to look at family and neighborhood level associations with early childhood outcomes. I spent time with a librarian geocoding and linking StatsCan administrative data, as well as Calgary Police Service data. And we computed the Vancouver Area Neighborhood Deprivation Index, or VANDIX, as well as an aggregate for neighborhood disorder using community crime reports. And we had a subsample of about 2,400 participants for whom neighborhood data during early pregnancy could be geocoded, and that was across 192 neighborhoods. And we looked at first was infant sleep at 12 months. And after accounting for family level factors such as household income, breastfeeding, and co-sleeping, our multi-level modeling suggested that neighborhood disorder and maternal perceptions of unsafety in their neighborhood, both during pregnancy, predicted less consolidated sleep. So that's the 2 solid lines here. And then in the diagram on the right, we also found associations between the association between deprivation and between neighborhood disorder and safety were mediated by maternal anxiety. So maternal anxiety could also potentially be another modifiable factor that we could target. Okay. And then with an honor student, we looked at overall development as measured by the Ages and Stages questionnaire, so the ASQ in the diagram here, which is a parent report on 5 different domains at 5 years old. And what we found was neighborhood deprivation was directly associated with poor development, overall development across all 5 domains at 5 years. Disorder was not directly related to the ASQ, but we did find that disorder predicted a positive slope between parent-child reading and development total scores. So put another way, as the number of crime reports increases in a neighborhood, the association between parent-child reading and general development gets stronger. So we could conclude that parent-child reading becomes more important when there's more disorder in a neighborhood during pregnancy, thus could help buffer potential negative impacts. And I forgot to mention I'm a proud auntie. This is my sister and her son, Jackson, in the photo. And then COVID-19 hit, and we all had different experiences of what happened there. And a team of researchers at the Alberta Children's Hospital Research Institute got together. This was led by Jerry Giesbrecht, Catherine LaBelle, and Leanne Tomfor-Madsen. And we launched the National Pregnancy During the Pandemic cohort study to see what was going on with expectant parents during this global crisis. And I was grateful to be invited to help put together the online survey. We spent many long nights on Zoom in April 2020 getting that together. We were able to recruit over 10,000 pregnant individuals across 1 year. And as you can see from the map here, we had coverage throughout the whole country and oversampled in some of the larger urban areas. And we had surveys that were sent out each month during pregnancy, then at childbirth, 3, 6, and 12 months postpartum, and more follow-ups are underway as more funding comes our way. Okay, so I wanted to show you some data from the roughly first 2,000 participants of the survey that have been analyzed and published. which unsurprisingly, but still alarmingly, showed significantly elevated levels of anxiety and depression symptoms. So we have 57% of clinically elevated symptoms on self-report scales for anxiety in the blue dot here, and 37% for depression symptoms over clinical cutoffs for depression in the red dot here. So this is very high compared to pre-pandemic rates, which we can see in the green bars are around 20% for anxiety and 10% for depression in green. What's potentially modifiable here, since we can't change COVID-19? Well, there is a good news story. So I wanted to show you another important study from a postdoc on the team. I wasn't directly involved in this paper, but it has informed our work, and it points to social support as a potentially modifiable factor. So they conducted diffusion tensor imaging with 58 babies when they were 3 months old, just out of Calgary, to assess early brain development. And the graphs here show connectivity between the amygdala and 2 different areas in the prefrontal cortex. And what we can see by looking at the purple lines is that when there's higher maternal distress, so exposure on the x-axis increases, that was associated with weaker functional connectivity, which is modeled on the y-axis. But the blue lines show us there's no association when social support was high, so we could conclude a protective effect. So we want to target social support. We looked at another potential risk factor, and I was involved in this study. We looked at what could be related to this increase in mental health symptoms that we were seeing during the first 4 months of the pandemic. So we used latent growth curve models, and what we found, if it will click. So we found that mental health symptoms did decrease over the 4-month period, both for anxiety and depression, although they remained relatively high compared to those pre-pandemic levels. And we were interested in sleep. So we saw that sleep duration and self-reported sleep disturbance actually increased over time. And then we looked at the relation between the two. And here we saw an association between the sleep intercept, so baseline, and mental health slope, so change over time. So poor sleep, that is shorter duration, more disturbance, and more sleep-related impairments at baseline predicted slower decline in anxiety and depression symptoms over time. And increases in sleep-related impairment and disturbances over time, so the sleep slope, also predicted a slower decline in mental health symptoms over time. In contrast, rates of depression and anxiety symptoms were not predictive of changes in any of the 3 sleep variables. So these findings suggest that detecting and intervening early in pregnancy to target sleep problems can be important for optimizing mental health. So now I will go into how this work can be used to inform interventions with a specific focus on e-health, so anything that is web-based or utilizing technology. And we have decades of research that demonstrate that matching individual preferences predicts increased likelihood of active participation in programs, which can improve treatment effects or positive outcomes for participants. And there was some very interesting research priority setting work that came out of Alberta with parents from conception to 2 years, and they identified their top priorities as support for families to develop healthy coping and emotion regulation, and another priority was sleep. And through this work, as well as some parent advisory focus groups, families also identified wanting access to evidence-based information that's tailored to their needs and delivered in timely formats. And some other parent preferences work showed that most parents prefer connecting with other parents, and actually 70% would prefer to do that anonymously. So we're seeing that there's a desire for this type of information and for it to be delivered potentially through e-health, which could fit the bill. So evidence does suggest that e-health programs can improve the mental health of parents of young children. So I got to lead a team of stellar students from the University of Calgary and University of Manitoba in conducting this meta-analysis. We included 38 studies across 4,300 participants from 13 countries, published between 2000 and 2020. It included a range of types of programs, from apps, websites, using phones or video conferencing, or a mix of these. And results indicated that e-health interventions were associated with better self-reported mental health among parents. And this was parents of children from 1 to 5 years, so first 5 years of life. And this was regardless of study design, so whether it was controlled or just pre-post, and also across most study outcomes. So we did some sub-analyses and found this held for anxiety, depression, and parenting stress, all with small to medium effect sizes. So e-health interventions offer promising and accessible option to promote mental health among parents of young children. So we capitalized on this, and one of the interventions I got to be involved with during my postdoc, targeted sleep, which we saw was a risk factor for mental health. So cognitive behavioral therapy for insomnia has been shown to be quite effective in adults in the general population, but there's less research during the perinatal period. So our team adapted it for pregnancy, and you can find a description in this book, a shameless plug. But the intervention consists of the 5 core elements of CBTI. And what's different is that we start with an understanding of sleep problems in the context of pregnancy, so how hormones and physical changes can impact sleep, as well as the family environment. We try to use pregnancy-specific examples and psychoeducation. For sleep restriction, we do use this, but we limit it to 5 hours to avoid any potential harm to fetus, and we're flexible with childcare or feeding at night. For stimulus control, that is used as well, and we focus on potential discomfort related to pregnancy, back pain, frequent urination, those types of things, as well as other childcare disruptions, maybe using baby monitors, for example. Many pregnant participants have other children. And a lot of the cognitive restructuring focuses on fears about how sleep might impact the developing baby. And then in the final session, we talk about infant sleep and the importance of mutual regulation and preparation for bringing baby home after childbirth. So we conducted a randomized controlled trial. You can see the protocol here. We recruited a community sample of women with insomnia who were 12 to 18 weeks pregnant, who had to score above a cutoff on the Insomnia Severity Index and met criteria on the SCID for sleep disorders for insomnia. And we had about 30 participants randomized to CBTI and 32 to a treatment as usual waitlist group. And I'll note that we started this just before the pandemic. So 5 participants received their individual therapy in person, but then we had to switch to online delivery during the pandemic using the Jane platform. So then we got to test if this worked with telehealth, and I got to join as a trial therapist at that time. And we did pre-post testing with self-report symptoms of insomnia and sleep problems, as well as a sleep diary and wrist actigraphy to measure some other sleep parameters. So I'm going to show you some unpublished findings. It's under revise and submit, so please no screenshots for now. We conducted multi-level modeling in R and observed significant time by group interactions. So this way you can see that I circled here. So participants who received CBTI had larger decrease in their self-reported insomnia symptoms and sleep problems at T2 is post-intervention and T3 is follow-up. And secondary analysis revealed no differences between the in-person and telehealth groups, although there was only a small group that got in person, so it's an unbalanced analysis. Not depicted here, but we also tested and found that fewer insomnia diagnoses were observed among the group that received CBTI than the treatment as usual group at both post-intervention and follow-up, indicating more remission with treatment. Interestingly, we did not find any statistically significant treatment effects for the actigraphy or wristwatch measures on any of the sleep parameters. I'll note that these were averaged across 7 days at each of the 3 time points. Inconsistent or mixed findings have been observed for actigraphy in other trials of CBTI with adults, and they suggested it could reflect poor differentiation of quiescent wake from sleep, so it might still be tracking them as awake. And potentially some missing data could be influencing that as well. Similarly, there was a lack of treatment effects for the diary measured outcomes, except for onset latency, where participants who received CBTI saw a decrease from baseline to post-intervention. So they reported they needed less time to fall asleep than those in the waitlist group. Okay, and then I'll quickly move on to another intervention. So shortly after the pandemic and seeing these higher rates of mental health problems, my postdoc supervisor, Leanne Tomfor-Madsen, collaborated with Leslie Roos from the University of Manitoba, and the BEAM team was born. And we tried to develop an app-based intervention that would target both mental health and parenting difficulties that could be rolled out rapidly and widely to reach families in need. So this program was 10 weeks. There's different modules on the app that are released each week. The mental health component utilized trends diagnostic treatment for emotional disorders. And then we had emotion-focused parenting strategies matched up with that. We also had an online forum. It was third-party software where participants could chat with each other anonymously. We added a telehealth group to review content every week, just in case there were some, tech problems. And so there's access to clinical coaches and peer coaches as well. So previous participants of other studies. And we conducted a pilot randomized controlled trial with mixed methods. So we had participants randomized to either the BEAM program or treatment as usual. You can see in the BEAM group, we did lose quite a few participants to follow up, and that mostly related to technology issues. We gave a lot of questionnaires. We created an aggregate measure for maternal mental health, an aggregate measure for parenting problems and positive coping. We also measured some secondary outcomes. We used the child behavior checklist for child outcomes. And because it was a pilot, we also had some measures of feasibility and acceptability. So a self-report questionnaire, as well as analyzing some of the back-end data of the app and conducting focus groups. In terms of feasibility and acceptability, engagement was relatively high at the beginning of the program, with 78% starting BEAM, so creating a user account. But as you can see from the table here in the top part, about 70% attended at least one telehealth session and participated in the forum. We had a lot watch at least one video. And in the bottom part of the table, we can see that most respondents felt satisfied with the app and found it easy to use. In terms of the main outcomes, we conducted mixed models with intention to treat approach, and the statistically significant results are bolded here. And I'll just draw your attention to the time by group interaction that indicates a treatment effect for overall mental health, that composite we created. We also looked at the individual scales and separate analyses and found greater reductions in anxiety and sleep among the BEAM group compared to control participants. There were moderate but not statistically significant effects for parenting problems and positive coping. We had good effect sizes at 0.36 and 0.41 respectively. And there was a moderate but not statistically significant effect for the child behavior problems. That Cohen's d was 0.41. It was likely that we were underpowered. Also, by accident, the treatment group might have acted as a control or an active control because they also got the weekly surveys for depression and parenting stress. So some self-monitoring could have been at play there. Oh, and in terms of qualitative results, there is a separate paper under review with a full thematic analysis of participant experiences led by a student at UManitoba. But I'll show you just some of the quotations that we got from our open-ended questions. So data about the videos show that they were mostly helpful and interesting. However, there were some technical difficulties about them not being able to play or skipping on some devices. We had some mixed findings for the forum. Some found it was their favorite way and were able to connect with others, and some found it difficult to read, and they were not able to comment on other people. You can't reply or add little thumbs up, little things like that. And then the telehealth sessions had pretty high acceptability. Participants emphasized the benefits of social connection, sense of community, and reviewing strategies. Others felt like maybe the timing could have been used differently for that, but overall, these results are promising, and we use the feedback to improve. And some of the participants joined as parent coaches. We launched Beam 2.0 after this using rapid cycle iteration. So we had a second randomized controlled trial with a bigger sample of 140 participants and slightly better follow-up or not as much attrition this time. And then in terms of engagement and adherence, we had 94% log into the app or attend at least one group session. And on average, participants attended about half of the telehealth sessions and watched a third of the videos. Forum participation was relatively high, but short in duration. In terms of the main outcomes, the significant results are bolded here, but I'll draw your attention to the treatment effects we found for anxiety and anger. In particular, the overall mental health composite was significant as well. And then for parenting, in particular, we saw reductions in harsh parenting. So together, the findings we have so far support the effectiveness of Beam as an innovative app-based intervention for improving mental health and parenting. I'll mention it was for parents whose children were 18 to 36 months, so during that kind of toddler preschool range, that can be very difficult. We had more engagement our second time around, and we did still find some areas for improvement. Oops. So Beam 3.0 is currently being rolled out with more professional videos, better forum, and some integrated symptom monitoring right within the app. Okay, so I wanted to leave some time to pass over to a trainee, Chantal Zorzi, who is a visiting MyTax Global Link Research Awards student, who will talk to us about one more intervention that we've been working on.
Hi, thank you for having me. I'll talk about my work in Dr. Anna McKinnon's lab, which is about using virtual reality for perinatal mental health. So next slide, please. So before we get started, I'll briefly talk to you about what virtual reality is. For those who don't know, VR is a 3D environment that someone can interact with, and it's engaging, can be motivational, and it's immersive. And for these reasons, it has been shown to be effective in treating anxiety and depression in a variety of fields. And my project with virtual reality is part of a larger project, which is called Toi, Moi Bébé. And Toi, Moi Bébé is an online platform, an online website developed by a team of researchers at the Saint Justine Research Center and Hospital to promote mental health and well-being of new and expecting parents. Essentially, it has 8 web-based modules, as well as telephone calls with coaches, and it uses CBT and mindfulness and acceptance-based skills as techniques. And it is soon being deployed through the government as well. So next slide, please. So my research objective is to try to increase perhaps the engagement of Toi, Moi Bébé through the integration of VR. And specifically, my research questions are 3. The first one is, in what ways can therapeutic VR techniques alleviate the current challenges in perinatal distress treatments? Second one is, how can VR therapeutic techniques complement online interventions such as TomoBB? And third one is, to what extent can VR therapeutic techniques help new parents learn practical skills for real-world difficulties? And to answer these questions, I have a four-step protocol. The first one, well, I had to gather the background information, and we did this through writing a rapid scoping review, and that is currently done, and we are aiming to try to get it published soon. The second step is a co-design workshop with parents in the perinatal period. We have already completed those workshops as well. The third step is the development of VR environment, which is a work in progress. It started at the beginning of this project, and it continues to go on until we have a good prototype that parents are happy with. And the final step is user acceptance testing, in which we will test this final VR environments. So in regards to the scoping review, we worked with a librarian from the Université de Montréal, and we searched 5 databases. We didn't find much literature. Most studies were published recently, with the first one we included being published in 2019. However, we saw lots of articles and protocols being published in 2023, so that is really promising for the future. It shows that this research is really starting now, and it's developing very fast. Regarding the virtual reality used in these studies, the stimuli, the environments that the participants saw were mainly in nature to promote relaxation and mindfulness, fetus and baby images to help get a better connection with the child, between the mother and the child, and mindfulness techniques and cognitive exercises. In terms of hardware, about half of them used standalone VR, and the other half used virtual reality in conjunction with a smartphone. And the results were overall positive. Participants were happy with their VR intervention, and they showed reduced symptoms of anxiety and depression. So in regards to our prototype development, on the next slide, please, we used the Unity cross-platform software program for the development, which is coded in Visual Studio, specifically C#. The ideas for the VR environments came from a literature review and multiple meetings with the TomoBB team. So now I'll show a video about the environments that were developed. So participants were first given the option to choose a language, because it was conducted in Quebec, so we gave both the language of French and English. There's also background music, but I don't think you can hear it in the video. Then we asked participants to first select relaxation techniques, in which we offered 2 meditation environments, one being guided and one being non-guided. So this guided one, we just chose a random environment that for most people it's relaxing, so beach environment. In the actual software, there is the background music of the waves, as well as a guided meditation that was already used on the TomoBB website. Then in regards to the other meditation environment, it was non-guided. So for that one, we had a 360 just image of the forest, and we simply asked the participants to look at the forest and hear the sounds of the birds and the forest. We also had birds flying, and just try to relax and feel immersed in the nature, just to really understand if participants prefer a more realistic environment or a more gamified environment, which one of the two they prefer, and which audio option they prefer if they want to meditate themselves or have perhaps a voice that guides them through it. We also had then 2 interactive environments. The first one, we asked participants to differentiate between helpful and unhelpful thoughts. So on that cube, which is attached to a balloon, we have different thoughts pop up. So we had different prompts. In this case, there were or in French, but they're the same ones for the English version. And participants, by touching them, they decided if they were helpful and unhelpful. Afterwards, after they were done sorting them out, they could liberate the unhelpful thoughts. So they were put in the side where they put all the unhelpful thoughts. They could grab the thought, look at it, and free it, and kind of let go of the thought. Then we have one more environment. They didn't do anything in this prototype with the helpful thought, just the unhelpful one. And for the final environment, it was more on the artistic side. So we had participants draw how their thoughts make them feel, or that's at least what we asked them to try and do. So we first asked them to try and draw how their unhelpful thoughts make them feel. They were able to change the color of the pen there, and it's a 3D drawing, so they could draw all around themselves. And then they also, in the next prompt, we asked them to kind of draw how they're helpful, unhelpful, how the other kind of thought makes them feel. And after they were done with their drawings, a little prompt came up, which asked them to observe both of their drawings, so they could both see their drawings. So regarding the results after showing these environments, on the next slide, thank you. We conducted these co-design workshops. We had a total of 6 workshops with 13 participants. They lasted between 2 hours and a half. And we had this workshop in 4 steps. So in the first step, we asked participants to write on a blue post-it or their struggles that they were facing throughout the perinatal period, and some of the thoughts they came up with were anxiety, isolation, depression, the unknown, and sleep. And we are hoping that these prompts can help us come up with more specific thoughts in the environment for the thoughts option. And then we asked them to write in a pink post-it what helped them throughout this time. So such as meditation, mindfulness, sports, and social supports were some of the most common answers from the participants. Then after they wrote their thoughts down, their experiences down, and shared them with other participants, they were asked to experiment with the technology, in which they tried out all 4 of our environments. And afterwards, we asked them to evaluate them, so to write down the negative and positive feedback on all the environments, to give us a better understanding of what they like and what they think might not be useful. And we also asked them to please suggest new ideas to see if they were perhaps interested in any other activities. And finally, the last activity of the workshop was to share their ideas with the other participants and with us, the researchers. And just some of the feedback that we are starting to analyze now. So from the main menu, it was overall easy to understand and to navigate. Perhaps participants would like more color to make it more fun and more visually pleasing. Regarding the meditation, they would like more choices of environments and audios, and to be able to mix and match, because an environment that might be relaxing for me might not be relaxing for someone else. So just really giving them the option, and also whether they want it guided, non-guided, whether at what time they want the guided meditation to start and to stop, and also perhaps some small interaction with the animals in the forest would be useful. Regarding the activity of separating the thoughts, so because it's a bit more engaging, some of the participants struggle to understand how to interact with the environment, especially participants that had never done VR. So a visual tutorial would be more useful instead of just writing what they have to do. And they would also like to interact with their helpful thoughts after sorting them out as well. And regarding the drawing activity, overall, they were happy with the art side of things, but they would like more colors and different types of art as well. And yeah, those are the preliminary results from our workshop. So in conclusion, regarding the literature review, we have very positive results on how VR could be used as a treatment option for perinatal mental health. And about our workshops, our participants were eager to try VR and see the potential of using it as a treatment method, and they gave us a lot of feedback on our project and our environment. And participants do believe that VR could be also a fun way to engage their non-pregnant partners, especially men who perhaps like to game more and do not like therapy as much. So it would be like a fun way to engage them with therapy without explicitly having to talk about their feelings. So it's more to their liking. And participants also enjoyed the workshop and they found our study interesting. So now we have to improve the trauma VR based on the workshop results. And the next step after improving this VR environment is to do the user acceptance testing, in which we plan to invite back the participants, have them try our next prototype, final VR environment, and interview them about what they thought and administer some questionnaires based on VR and the technology. And hopefully in the future, continue with the project to the point of doing a bigger pilot study and randomized control trials. So thank you for listening.
Thank you, Chantal. So I'll just wrap it up there. Overall, this work aims to provide accessible, evidence-based supports to start happy and healthy families. We have all our references there. I'd like to thank everyone who's involved, from all the teams, funders, and of course the families who participated in the studies. And we'll see if we have some time for questions.
Any questions? Anna?
Yes.
I do actually have a question with respect to MWBB and being offered. So it's going to be deployed through the government. Can you tell me a little bit about this?
Yes, so this morning we had our big announcement with the Quebec government. Their press releases should be going out. We had some CBC Global News folks were there. They did it in French and in English. So here in Quebec, it was part of the Stratégie numérique, where they're putting different evidence-based programs online, accessible to the public. So it's accessible in Quebec now, and I believe because it's online, anybody can go log in and make an account and sign up. It's available in French and English. It's a slightly different version than in the research study. I'm not sure if the clinical coaches are going to be available, and some of the screening is a bit different, but there will be some data that's being tracked, like back-end data and maybe some provider data, so we can see on a larger scale what's going on. But that's all contracts with the government. So very exciting today. We can announce officially. You can go look it up.
Fantastic. Marilyn, can I have you ask your question, please?
Sure. And I'm sorry, I feel like I'm taking up too much of the oxygen for these questions, but I couldn't help myself. You have such great speakers and great topics. And so today, actually, I just want to make one thing, but in comment to your work, Anna, and it relates to what I said earlier to Rachel. We now are doing a much better job of monitoring the kinds of behaviors and phenotypes that you are alluding to, and I really resonated with your point about how actigraphy is, maybe not the best way to go, because it's like even just a step above questionnaires. You're asking a question about somebody's sleep, about somebody when they're sleeping or they're trying to remember how they slept. And then with actigraphy, just as you said, it could be anything. It could be you're not moving, but that doesn't mean you're asleep, and it doesn't mean you're in those sleep stages, which are so important in talking about sleep health. So I just wanted to mention, both for your work and Rachel's, finally, the wearable device manufacturers like Garmin and Fitbit and others are beginning to play in the sandbox with researchers. Before, they didn't want to share their raw data, but now they're realizing, oops, maybe we got to do that. We got to catch up with Apple and other, whomever. But so we are working with Garmin devices to measure sleep during pregnancy. And we're doing it at a couple of places here in the States and in Africa. But I do think it provides a low-cost, feasible way to assess, get a lot more useful, I think, information. It tells you a lot more, one, about sleep, but it also tells you about the autonomic nervous system, which is what Rachel's work made me think about. So I do believe it's worth taking a look at. Happy to chime in as necessary, but you could probably find out everything you need by reaching out to maybe Garmin or the people who have used the Garmin devices, because they're relatively inexpensive. You know, they're $100 versus some of the things you can use are ridiculous. So anyway, sorry, I will shut up there and just let you know that I think it would really maybe help in adding to really everything you're doing, including ways to document these wonderful clinical trials you're doing to look at, well, have you actually intervened with a modifiable risk factor? So thank you for what you guys are doing.
Quite well taken. One of the Beam trials did have Fitbit attached to it, and so no pun intended, some students are looking at that, and we are hoping to put in a grant to get some Muse headbands for moms and little ones and see what we can find out with that.
Yeah, great. Good.
Marilyn, can I ask you to ask your question, please?
Sure. Thanks, Michelle, and thank you, Anna and Chantal, for a really interesting presentation. So my question is more to do with the Beam trial, and I was wondering, because you highlighted, Anna, that a lot of the research that's been done in this area primarily focuses on women with the assumption that they're cisgendered women. So I was wondering what the diversity of that sample was, and if in that qualitative data you were able to, if it was a diverse sample and you included multiple parents and caregivers from different backgrounds, if you had any questions about what their experiences might have been. Just curious about the learnings that we can take from how best we can support diverse families in this work.
That's a very important question, and I don't have the demographic data with me for those first two studies, but what I can remember from our team meetings, there's actually a large percentage comparative to most studies from Indigenous communities in Manitoba. And so there are some Indigenous students and collaborators now working with that team to look into some of that data, of course, following proper ownership and data analysis approaches. And there are a few team members who are also interested in including partners in our next rounds as well. We tried to do a Beam Buddy in our first program, our first launch, and anyone that you were interested, that you felt close, whether it was a partner or friend or someone that could help you be involved with the material. It didn't really go over well, so we're exploring different ways, maybe adding a module for partners to join. And then I'm hoping that my team can start some priority setting work with gender diverse and queer parents. I think one student is looking at that right now, and I'm hoping to add a perinatal version. So expecting, and you're in that planning stages of becoming a parent. So that's also very important for us to continue with that work as well.
Thank you so much, Marilyn. Thank you, Anna. And Chantal, thank you so much for your time. Does anybody else have any questions? Okay, so we're just 1 minute over. So I would like to take the time to thank all the presenters today. But Anna, thank you so very much for your time. And Chantal, thank you very much for your contributions. We really do appreciate your participation. Again, if anybody has any questions, would like to present in the cluster, please contact us at wcgrants@ualberta.ca. Thank you so much for your time. We hope you have a great day.